Hello TYPSC students, welcome to the virtual classroom. Our today's topic will be the continuation from the previous topic. In the previous topic we have already talked about the pre and exo-erythrocytic Shizogony. In this part we have already seen that how sporozoites, first of all how the blood meal takes place, then sporozoite enters into the human blood, it goes towards the liver, and from liver or within the liver it enters into the hepatocytic cells. Once they enter the hepatocytic cell, their elongated structure changes into somewhat spherical structure or chisel formation takes place. After the chisel formation, they convert into cryptozoites. Cryptozoites, we have seen that the nucleus divides into many number, and then suddenly they come out of the hepatocytic cell. When they come out of the hepatocytic cell, they first called as cryptozoites, but later then second time they enter and go through the same process. And then when they come out of the hepatocyte cell, then they are of two types. One type is called as macro-metacryptozoite and another type is called as micro-metacryptozoites. And we have already seen that macro-metacryptozoite infects the liver cell again, and micro-metacryptozoite goes towards the RBC. Now here onwards we will start with today's topic, that is the erythrocytic Shizogony, means when the micro-metacryptozoite enters into the RBC. So when after the finishing of exo-erythrocytic Shizogony, micro-metacryptozoite enters the RBC, and it normally takes 8 to 12 days after the two phases, completion of the two phases, and the erythrocytic Shizogony starts. And during this erythrocytic Shizogony, there are several stages that takes place. The first one is the trophozoite stage. Trophozoite, we have already seen the ultrastructure of trophozoite. So we have seen the ultrastructure of sporozoite in previous lectures, and again the ultrastructure of trophozoite. So the trophozoite, once the cryptozoite enters into the bloodstream and fed on erythrocytes, it becomes rounded, modified into trophozoite. We have already seen trophozoite structure in the nucleus, mitochondria, different organelles are present. So in this stage, once the conversion takes place, the second stage is the signaling stage. So what happens, previously I have told you that inside the RBC they start eating the hemoglobin, and when they ingest the hemoglobin, it decomposes into protein and hematine. Okay, protein is a food source which is absorbed by the parasite. Okay, unused part, that is hematine, which gets converted into toxic yellowish brown malarial pigment, hemozoin. So hematine is converted into hemozoin. See, also during this process a very significant thing happens. That stage or that appearance also suggests that the person is suffering from malaria. Now this particular stage is called as a signet ring stage. Why it is called a signet ring stage? Because as the sporozoite grows, vacuole appear at the center of the nucleus, at the center, and because of this, the nucleus is pushed towards one side. Because it pushed towards the one side, it looks like a signet ring. It gives a ring-like appearance, as you can see in the diagram. So when you prepare the blood smear, okay, the person who is suffering from malaria or symptoms like that of malaria, that particular person's blood will be tested, and in that not necessary antigen testing will be carried out. First of all, the signet ring stage identification will be carried out. So we will make this smear on this slide, and when the staining will be carried out, at that time, if such a stage is observed, signet ring stage, then it confirms that the person is suffering from malaria. So this stage is called as the signet ring stage. Further, what happens when the signet ring parasite grows? Vacuole disappears. So in signet ring stage, you have vacuole. When it further grows, vacuole disappears and parasite becomes amoeboid in appearance. It looks like amoeboid. Okay. So thrusting out the pseudopodial process. So this stage is called as amoeboid stage. So initially signet ring stage, then you have the amoeboid stage. Okay, and during this stage itself, numerous granules are observed. Okay, and these granules are called as Schaffner's granules. So you should know this. Again, this is after the signet ring stage. This is also a specific stage that occurs in a malarial patient blood. Okay, so Schaffner's granules will be present in the amoeboid stage. After this, you have another stage that is called as a Schizogony stage, where parasite grows in size, become rounded, and almost completely fill the RBC with Schizogony. So what is there in the RBC now? Only Schizogony. And hemoglobin they have used already. Hematine formation is there, the toxin formation has already occurred. So even though the RBC is there, the total busting haven't occurred, but Schizogony stage is there. Now after this, there is the last stage that is called as the rosette stage. This whole stage or part is the Erythrocytic Shizogony. Remember, this everything is happening in the RBC itself. So this rosette stage is also in the RBC. So nucleus of Schizogony divided by multiple fission to form 6 to 24 daughter nuclei. Okay, and these nuclei are arranged at the periphery, while the toxic hemoglobin granules accumulate at the center of the RBC. Remember, as you can see in the structure in the diagram, you cannot see 24 daughter nuclei, but you will see the accumulation of the pseudopodial carrier at the center of the RBC. Now it appears as a flower rose, because at the center you see that yellow pigment, and around it you may see the nucleus. So it looks like a flower-like appearance, rosette stage. That's why it is called as a rosette stage. So nuclei is surrounded little cytoplasm. So remember, within the RBC, the number of nucleus is present, and at this stage, at the rosette stage, what is happening? That particular hemoglobin granule is present at the center, and around it many nucleus has been observed with little cytoplasm. The cell haven't separated yet. Okay, so when the RBC rupture occurs, after this the RBC rupture occurs, and these sporozoites are liberated into blood plasma along with the toxic hemozoid. Now remember, this toxic hemozoid, the malarial person symptoms is the shivering. Okay, it will increase in the temperature, you will have the shivering. This thing happens because of the increase in the presence of hemoglobin. Okay. So normally it takes fresh RBC to repeat the erythrocytic cycle. Now after this what happens? Merozoites comes out again to start infecting the RBC. So erythrocytic stage goes on. New merozoites will come out. It will start infecting the RBC. So what happens here? We will start the cycle again unless and until they are transferred to some other transfer to next stage. Okay. Now what is the next stage? That is the post erythrocytic schizogony. Now what is this post erythrocytic schizogony? Now merozoites produce erythrocytic schizogony, cycle is going on, and some of them reaches to the liver cell again and undergoes schizogony development of the liver cell itself. This is called as a post-erythrocytic schizogony. So, what is happening here? Some merocytes produced in erythrocytic schizogony reach towards the liver cells and undergo schizogony. There is some possibility that once they are entering into the liver RBC, at that time there is a possibility that they will start entering the liver cell. So now already within the liver cell, the cryptozoites, they are infecting, and now again these merozoites, they are again infecting the liver. Sometimes they remain within the liver for a longer duration, as I have told you the latent infection. And I also told you that many times this has happened. The person has been cured from the malaria and still they show the sometimes after 1 year or 2 years of infection, he may show the similar kind of symptoms. Why? Because some of the merozoites are present in the liver cell. Now, after this many cycles, then there is a change that occurs within the schizogony. Now that change is the sexual cycle of Plasmodium in man. Now generally the whole of the asexual cycle takes place within the man and sexual cycle takes place within the mosquito. But initially gamete formation may take place within the man. So that is called as a gametogenesis. So after many generations of about 4 to 5 in the blood, some merozoites increase in size to form 2 types of mastocytes. Now remember, this is very important. 2 types of gametocyte formation takes place. Okay. So merozoites forming first of all macro gametocyte. Size may vary from 10 to 12 microns, and these macro gametocytes are female. Okay, they are around with food-laden cytoplasm and small eccentric nucleus. So their nucleus is small. Remember, these are macro gametocyte because their size is large, but they are having small nucleus. In microgametocytes, the second thing that is formed is the microgametocyte, which is the male gametocyte. Okay, and they have a clear cytoplasm and a large central nucleus in that. So large central nucleus reason, and once this gamete formation takes place, now these gametes goes towards the peripheral blood system. From this peripheral blood system, blood or these gametes, they will enter into the some of the capillaries, and as we know, when the mosquitoes start sucking up the blood, at that time what happens here? At that time these gametes, both macro and micro gametes, they enter into the mosquito, and from there onwards the next cycle start. This is called as a sexual cycle of Plasmodium in the mosquito. Up to that, there is no sexual cycle. Within the human, everything asexual phases takes place. Only one thing that happens is sexual gametoformation, in which male and female gamete formation takes place. And after the gamete formation, macrogametocyte and microgametocyte. Remember, macro is the female gametocyte and micro is the male gametocyte. Even though macro gametocyte is a female gametocyte with a large size, but its nucleus is small. Whereas microgametocytes, these are male gametes, have a clear cytoplasm and large central nucleus. So this you should remember. This is the end of the asexual cycle, in which we have seen the pre-erythrocytic cycle, exoerythrocytic cycle, erythrocytic cycle, and post-erythrocytic cycle. And at the end, gamete formation takes place, and during the blood meal, these gametes from the infected person, remember, gametes from the infected person that enters into the mosquito stomach. Okay, so from there onwards, we will have another stage, that is a sexual stage. A sexual stage is over and sexual stage. Thank you.